So, hello, I'd like to welcome everybody that's joining us tonight to this discussion back to here at Breaking the Consensus at Protect Life. Before we start, I'd just like to bring your attention there to the old subscribe button. So if you can click on the subscribe button, that means that all the new material and all the new work that we're doing will be automatically adverted to that fact. Also, we're involved in a lot of campaigns, one or two very important campaigns in particular that you'll be aware of in Ireland, which with or without a pandemic, we have to go on with our work, and we need your help to do that. So if you can click on the donate button, anything that you can give will be really useful to us, and we can guarantee will be used carefully and well in our campaigns to defend the value of life. So tonight I'm delighted to welcome back Dr. Tara Sander-Lee from the Charlotte Lozier Institute in the United States. Charlotte Lozier Institute is a very prominent, highly respected pro-life institute interested in the scientific issues around the science around many bioethical issues. It's based in Arlington, Virginia, but Dr. Sander-Lee is joining us from snowy Wisconsin tonight because, of course, with the pandemic, we're all staying at home and moving as little as we can. So tonight what we're going to do is we're going to be talking to Dr. Sander-Lee, particularly about a lot of the concerns that people have around the issue of the vaccines, which, thank God, are starting to roll out in the midst of this terrible pandemic and giving us a little bit of hope. But there are ethical concerns that a lot of people have, and scientific and medical and others. So we want to address some of those tonight. So thank you again, Dr. Sander-Lee, for joining us. Welcome.
Thank you so much. It's a pleasure to be back, and thank you so much for the invitation.
Okay, I suppose the first one we want to kick off with is, particularly in the context, as I said, the Charlotte Lozier Institute, very highly respected and you coming from fundamentally a pro-life perspective. And I think one of the issues that a lot of people have concerns with is how ethically can we as pro-life people use these vaccines? Are there vaccines which are, shall we say, more morally acceptable or less morally acceptable? There are issues about the use of whether is it fetal tissue or fetal cells or cell lines and that kind of thing. So listen, I'll throw it over to you. Can you give us a general overview of individual vaccines and then the technology behind it and the ethical questions involved?
Absolutely, and this is such an important question, and we have been researching this in great detail in order to really understand how these vaccines were made. And so when we first started this, we looked primarily at the vaccines that were being developed and that were candidates. How were they being produced, and how were they being produced, and what was going to actually be injected into each person? So when we first did this, we looked at all the candidates, and we found that there were definitely a handful of them that had chosen to use human cell lines from past abortions in order to make their vaccine. And some of these that you can go onto our website and find this chart, but what you will see is that some of the companies such as AstraZeneca and Johnson & Johnson that we know are coming down the pike, they're not approved now, but these have chosen to use these aborted fetal cell lines to make their vaccines. And the use of fetal cell lines to produce vaccines is reason enough for some people to second guess a vaccine or avoid it altogether. As more information became available, we started to look more closely at whether they use fetal cell lines to develop or test these vaccines. And what we can tell you is that the really good news is that a lot of the COVID-19 vaccines that are candidates and that now are approved. So for example, the Moderna and the Pfizer vaccine are not made with any fetal cell lines. They use a completely cell-free system. It's called the mRNA platform. And so the mRNA is like a recipe, and it's like you're given a recipe for how to make the protein, the antigen that will mount your immune response, but it will not, but there are no cells involved in making this vaccine. So that's the really good news, that a lot of them have chosen to use another platform that doesn't involve fetal cell lines. But as I said, a couple of them still do. And we do know that some of them have chosen to do some testing separate from production. And we know that this is controversial too. And we want to see all the use of all fetal cell lines come to a halt. We don't want any manufacturers to use these fetal cell lines, and we urge all vaccine developers to avail themselves from the many of the many alternative options that do not involve human fetal cell lines. But we do think it's good news that that the two approved here in the United States and that are approved there in the EU, the Moderna and the Pfizer, are not made with fetal cell lines.
So just for clarity on that, because I think a lot of people want to be reassured, there may have been some fetal cell lines used in testing the efficacy of these vaccines, Pfizer and Moderna, but in the production of the vaccine, and the vaccine that the person is receiving will not contain any material which is derived from an abortus fetus. Is that correct?
That's correct. That's absolutely correct.
So, what would you say to somebody who says, well, okay, it doesn't contain any, but they did use it in trials, they used it in testing, and I don't like that. I mean, that's an understandable reaction, but you think that maybe that in the context of a pandemic, we have to maybe stand back and understand the situation. Shall we say it's probably good?
Yeah, I mean, and this is where it becomes really, really difficult, because the fact that some of these developers have unnecessarily put families in this difficult position by choosing to use the human fetal cell lines, whether in development, production, or testing, this really places families in a difficult position of what they should do. Because we know that alternatives are available, and they had alternatives available to them. And we know this just because already we know that vaccines that are currently available have chosen to use alternatives. But what Charlotte Lozier has done is we do not take a one-size-fits-all approach to how people should respond to the controversial vaccines that use fetal cell lines. Rather, we provide and will continue to provide the most accurate information in a timely fashion, so that so people really understand specifically when they go in to get vaccinated, they can be prepared, they can be informed and well educated, knowing exactly what was done with that vaccine, how it was made and how it was tested, and then weighing all the factors, including the availability of non-controversial vaccines. Then we believe that the judgment is best made by each individual and their family. Until the vaccine industry ceases the use of all fetal cell lines, unfortunately, we are in this position where we have to make some really difficult decisions. And for some, that decision will be, I don't want to take a vaccine that is made with fetal cell lines. And it will be pretty clear cut and dry, right? And for that, for those individuals, then they will feel they will feel comfortable taking the Moderna and the Pfizer vaccine. There will be others that will second guess even the use of using fetal cell lines for testing. And we know that that's a difficult decision for them too, but at least they'll have all the information. The good news is that they're actually, it looks like CureVac is one company that right now we have evidence that they have not used fetal cell lines at any of the 3 steps of development, production, or testing. So there is the possibility that there might be some options in which they didn't use any fetal cell lines. I mean, the data's still coming in, and we're reviewing it as quickly as we can, and there's a lot of analysis to do, but I think there still is hope that as more of these candidates come down, become available, that there actually might be some options where there weren't any fetal cell lines used.
Can you just explain something for me, because I'm not as a layperson, I'm not clear on this. We hear a lot of talk about fetal cell lines. So when are we talking, when in the production they use fetal cell lines, or in the testing they use fetal cell lines. Are these cells taken from aborted fetuses?
So I want to first tell you what it's not. So a fetal cell line is not the same as fetal tissue. So you might have heard about all the controversy with the David Daleiden videos and the Center for Medical Progress showing the trafficking of all these fetal tissues happening happening now, right? So that's not what we're talking about. So a fetal cell line is derived from an abortion, which addresses your question that you just asked, but it is a cell from fetal tissue that was obtained decades ago. So in the case when we were talking about like the HEK293 cell line, that stands for human embryonic kidney. The 293 stands for the number of experiments that it took to actually make this cell line. It's not the number of kidneys that it took, it's the number of experiments. So what they did is there was an abortion, they isolated cells from the human embryonic kidney back in 1972.
Okay.
And then they established this cell line in 1973 that then they expanded these cells. They created lots of these cells that are then put into the freezer and then brought back out. They're used like mini cell factories to produce large quantities of protein. But they can be brought in and out of the freezer over and over again. And so they do the use of these cell lines, we don't condone it. We want all of this practice to stop. Anything that is remotely connected or directly connected to abortion is wrong. But I think it's a really important distinction that these cell lines do not require the ongoing destruction of babies.
Okay.
So hopefully that helps to explain that.
Cool. Yeah, I think that's helpful. So, but you said also, so it'll be interesting what you're saying about the CureVac. We will put a link in, I know Adrian will put a link in to the Charlotte Lozier site, because for those who haven't seen it already, there's a very useful chart there, if you like, which gives people who are concerned all of the information on the various vaccines. And I imagine you're constantly updating that as new information becomes available and new vaccines are under consideration.
Yes, that's absolutely correct. We continue to keep it updated, and which ones are the ones that are approved, we are keeping track of in the United States. But all of the information regarding, how they were made and produced, tested, and developed, all that is going to be the same no matter which country you live in and where you live. That's going to all remain the same, and that is being updated frequently.
Okay. We have a number of questions, and I don't expect that you're going to be able to necessarily speak directly to the question, but maybe you can speak to some issues of it, because obviously I know you're very careful not to speak too far outside of your personal magisterium, your own expertise. Some people have been concerned about, okay, well, lots of concerns obviously, but okay, one is that there's been a lack of transparency in the data. There was a letter in the British Medical Journal. We had a communication from a pro-life doctor here in Ireland who read it, and the British Medical Journal is BMJ is very reputed journal, one of the top medical journals in the world, and a former editor, I think, or associate editor, said, a lot of the data hasn't fully been released about the efficacy. Do you have any sense of, because you're looking at the data that's coming out, about the data, about the level of completeness, or if there is maybe an excessive amount of data being withheld? The FDA is not considered usually to be a rash approval body. In fact, many people criticize the Federal Drug Administration in the United States, which approves drugs, as being excessively conservative. And usually these bodies are fairly conservative, yet they have licensed these drugs, these vaccines. Mm-hmm. How do you see this issue regarding the data, the availability of data?
So, I will tell you that the data that is coming out, especially with those vaccines that have been approved, so Moderna and Pfizer, the mRNA vaccines, the efficiency of these vaccines is very high. The data looks incredibly good. I just want to give you some statistics. So early phase 3 trial data showed 94.5% effectiveness in preventing COVID-19 from Moderna. When we look at Pfizer, the vaccine was found to be approximately 95% effective. And it has received, both of these have received emergency use by the FDA in the US. And as you said, they definitely do a very thorough job of reviewing this data. I know in the EU, both have received conditional marketing authorization and approval following evaluation by the EMA. What I will tell you that I think just even in my own research, I have found it difficult to find the data for both of these on the EMA website. But if interested, people should be able to get access to the publicly available documents and data. So for example, we know that Pfizer has published their phase 3 trial data. I believe that we're still waiting for the publication from Moderna for their phase 3, but their phase 1 and 2 clinical trial data should be available publicly. But you can definitely get access to the publication data for their phase 3 trial data. As far as looking at other forms of data, you should be able to, and I apologize if you can't get access to the data by the EMA website. I'm not quite sure why there has been difficulty in getting that information if you want it, because it should be available. But you should be able to go to the FDA website through the United States, and it should be really similar data with regards to the efficacy and the safety. And if you go to the links at the top under COVID-19 vaccines authorized for emergency use under the FDA website, and then scroll to the bottom, you actually can see all the documentation from the Vaccine Advisory Committee meetings, and you can then go through those links from December 10th and the December 17th meetings and go to the events materials on those pages, and you will see all of the submissions as well as the analysis from the review committee. So if you are not able to get the information from your own country, from your own regulatory agency, you should be able to hopefully get it publicly from other sources. So hopefully that helps. So I really don't believe that there is any ulterior motive in trying to withhold data. And hopefully, if you have any questions on how to get some more of that specific data, be sure to reach out to me and the Charlotte Lozier Institute. We can help you find that.
Okay, just, I mean, for clarity, even if there is some data from the clinical trials which has not been made public, that doesn't, all of that data will have been made available to the FDA and to the EMA, won't it? I mean, it's not that there's information which they've held from the regulatory authorities. Even if there is some which is in the public discourse, all of the data will have had to go to the approving bodies.
All of it will have had to have gone through the approval bodies, and it has gone through the approval bodies in the United States and through the EU, through the EMA. And so it absolutely has gone under careful review before being approved for actual distribution for vaccination.
Now, we're in the case of Moderna and in the case of Pfizer. Again, if layman here speaking science, which is not a language I speak fluently at all, but this is a new technology. This is based on using RNA, right? Which I remember from my biology classes, many, which is not but it's just ribonucleic acid, whatever. Now,
right. Mm-hmm.
I have heard people express worries or concerns that in some way these vaccines can alter a person's DNA. Is this a valid concern, or is this a misunderstanding of the science?
I can understand why people are concerned, because it's a new technology, it's a new platform. This is the first time that an mRNA vaccine has been approved for use. But I can tell you that scientifically, the mRNA vaccines do not work by changing your DNA or your RNA. So they do not cause any genetic modification. So the mRNA is as you mentioned, is a nucleotide, and it's a recipe to make a protein that is going to be read by your cells. And after it's read by your cells and your cells do what it's supposed to do, which is making the spike protein so that you can then, mount an immune response to that target antigen. After that mRNA is read, it's going to be then discarded and it's going to be broken down. So it's not going to hang around. It's never even going to go, if you remember back to basic biology and cell biology 101, the mRNA, as it comes into the cell after you're vaccinated, it's not even going to enter the nucleus. It's just going to stay within that cytoplasm, be read. And then it's going to be discarded after it's used. So hopefully that helps to address that concern that is out there, that really is one that people shouldn't have to worry about.
I know this is a silly question because this is a vast subject, but we're talking 101 again, biology 101. Vaccines work by stimulating a response in the body to make the body respond to these foreign bodies, to make them better at recognizing them and killing. So this does it by stimulating the creating of a protein. This is a new technology. The older technology, which is being used, shall we say, more traditional vaccines, that's the vaccines, say, like in AstraZeneca, is that right? These are using the more traditional methods of vaccines, using what is adenovirus?
Adenoviral, yeah. So there's actually 5 different platforms being used to produce the different candidate vaccines. The AstraZeneca and the Johnson & Johnson, or sorry, Johnson & Johnson, also in combination with Janssen, those 2 vaccines are adenoviral vector-based vaccines, okay? So those vaccines need cell lines in order to express their target of interest. And in that case, they use an adenoviral vector to deliver their target, the spike, and then overexpress it in these. And in their case, they use the fetal cell lines. And then that's what they use to then that produced part of the viral protein, that's what they use as a vaccine. But then there's other forms, like the flu vaccine, for example, is actually an inactivated or attenuated virus that isn't functional, but then that's going to be attenuated and weakened, and then that's what's going to be given to people. So there are actually 5 different methods, and we actually have all of these methods well drawn out and illustrated on our website. And we can give you the link if you want, and you can put it on your website, but it goes through all 5 different methods. And yes, the mRNA nucleotide vaccine along with the DNA are 2 very new platforms.
But I know, because this is something we constantly hear, so maybe you can nail this just generally about vaccinations, but in this one in particular. Say, like in the flu vaccination, I very often hear people say, what it actually does is it gives you the flu. But that's not really true, is it?
No, it's not true. So it gives you, it will give you, what that vaccine is going to do is it's going to be the virus that is, like I said, weakened or inactivated, or there's going to be a part of the virus. It's going to be the virus that has been weakened or inactivated, or it's going to be a part of the virus that has then that isn't as functional. And all it's doing is that it's training your body to mount an immune response to that target, so that when it sees the real thing that is virulent, that will actually cause an infection, then your body will have already made the antibodies that will and the T cells that will then attack and then kill the actual foreign substance. So hopefully that helps to explain that when you receive a vaccine, you're not getting the actual virus. You're getting something that is going to kind of trick your body into developing an immune response to something that will recognize the real virus when it's actually seen.
But so to draw a line under it, you can be assured that there isn't a mechanism here that would allow these vaccines to in any way alter your DNA.
That's correct.
It just doesn't work like that. Okay.
Right. Right.
No. Okay. A lot of people, and I'd include myself in this. Now, in one way, we are massively impressed and grateful that
Mhm.
the speed at which these vaccines have been produced. Because when this pandemic comes on, everybody says, oh, well, if we get a vaccine, because it's not a guarantee, because lots of times we try and we don't succeed. If we get one, it'll be at least 2 years. And even then, that will be super quick, because normally it takes 10 years to develop a vaccine. And if polio took this amount of time, and this, so on and so forth. Here we are, we're basically a year into this pandemic, and we have all these vaccines. Suddenly, they're
Mhm.
there. Have they been cutting lots of corners? Have they been skipping steps that they should have been taken? I mean, is this is this a sort of a panicked response? We have to get something out, or have they actually been doing all the things they would normally do? And if they have, how come they've done it so quickly and what
Right.
normally takes so long?
Yeah, and that's a fantastic question. I mean, because the speed with which these vaccines have become available and have crossed the finish line is remarkable and unlike anything we have seen before. And so, understandably so, people are skeptical and concerned that corners may have been cut and that the final vaccine product might not be as good. I just want to explain that historically, and I think this is so important that you mentioned this, Michael, that the traditional process of vaccine development, as we just talked about, the attenuated or the inactivated vaccines, has historically been lengthy, right? They can last well over even a decade in some cases to make these vaccines and have them available to people. But the COVID vaccine development has been seriously accelerated, in part because of what we were just talking about, in part because of the technology. These novel approaches have really helped to drive rapid vaccine production. And we've seen publications of this that have, there's a Nature publication in particular that has explained this in more detail. But in particular, these mRNA approaches of Moderna and Pfizer, that, or BioNTech, the collaboration between those two, has really helped to drive this speed. And so it's also allowed them to initiate large scale production without assurance of success, as well as overlapping trials. So meaning that basically before they even knew that they were going to be beneficial, they could massively produce these in advance and have them ready to go. And then once the clinical trial, once everything was, once then there was assurance of success, then they were ready to distribute. So just in one example, if you look at the one publication where Moderna breaks down the timeline of their design and development and their production, and then and now what we know of as the clinical trials that have now completed. Once they knew the sequence of the SARS-CoV virus, and that was published actually earlier this year, and I believe it was in January. Once they had massive governmental funding support, they were able to initiate large-scale production of their vaccine just 4 days later. And then they were able to begin clinical trials just 2 months later. So that just shows that, you know, they didn't need the historical, you know, what they used to use to large scale, using these cell lines to produce their protein. Moderna was able to just basically sequence their mRNA, design their nucleotide sequence, and then initiate production right away. The BioNTech one was actually very similar. And testing was not curtailed. I think what we had talked earlier, that approval for public vaccination campaigns was granted after conclusive safety and efficacy testing became available and evaluated by the major national regulatory agencies like the US Food and Drug Administration and the European Medicines Agency, the EMA. So, and this, actually, if you look at some of the clinical trial data, you will see that the size of the trials, that is the number of people that they recruited to actually to actually be part of their clinical trial and test the vaccine, that actually, if we look at some of the largest clinical trials undertaken, COVID-19 accounts for 5 of the 10 most expansive in terms of enrollment. So for example, like Moderna looked at 30,000 people that were vaccinated. Pfizer looked at 44,000. And so these are actually really quite large clinical trials that were done, and all of that was reviewed and approved prior to anybody now being given the vaccine.
Okay, if I can synthesize and then you can correct me. One of the central things that's different now about vaccine technology is our capacity to map the gene, which was something historically people couldn't do. So there was an awful lot of time spent in that process of understanding how the virus, understanding the virus itself. Going back, I suppose the first time this was when the huge step was made when the virus, they mapped the virus for HIV. I remember that was this incredible, and that took an immense amount of time. Now, within a matter of days, they were able to actually understand the map, do the gene mapping of this virus, and that makes it a massively more efficient process of how you understand how you respond to it. So that
Yeah, and you're absolutely right. And I think the other thing to mention is that there were two other pandemics before COVID-19, right? SARS and MERS. And those are coronaviruses, right? They're kind of like cousins of the SARS-CoV-2, right? So there was a lot of research and development that was done on those viruses so that they understood how infection even occurred. They already knew the target. They knew that spike was the main target, what they needed to target. So prior to COVID-19, there was actually a lot of information that was already known from previous research on those other two coronaviruses. And that massively helped move things forward as well. Once they knew the sequence of this SARS-CoV-2 virus, then they were able to run because they already knew how the infection actually took place.
Now, in the past, if you're applying for approval for a vaccine, would it normally have been the case that you would run your sets of clinical trials for immediate reactions? So, for example, we know that there is a problem sometimes in a very, very small number of cases for anaphylaxis, but which is treatable. As long as, and that's one of the reasons why the person has to stay for 15 minutes. If there's going to be an anaphylactic reaction, it'll happen very quickly. People can administer, then they can respond medically, and that can be treated. So and even then, it's a very, very small number. That's an immediate reaction, and these trials have happened over periods of months. But in the past, would it have been the case that they would say, okay, there's no immediate, but you have to come back to us in 5 years' time with more data to see if there's long-term effects, and then we'll license you? Like what I'm saying is it normal that you would get permission, you get licensing after this period of time? Because we don't know. I know, say for example, I've heard people say that in the case of the swine flu, that people believe that maybe there was if there was an effect of the vaccine which happened a year or 2 years later, that there were long-term consequences. I mean, can you speak to that?
Well, I do know that even so I will like you mentioned, like there is the concern with anaphylaxis and allergic reactions. There are expected adverse events. We do know that these are new vaccines and for a new disease, and so rare events will appear, and then rare events will appear, and we know that they're going to appear. We know that but they're not common. We do know that the most common side effects is like going to be pain at the injection site and fatigue and the like. But when we're talking about these more rare events that we're hearing about in the short term, like we've heard of, like there have been some that have talking about Bell's palsy that was reported to be more frequent, but there's not actually a sufficiently large number of cases to conclude that this was beyond what would naturally be observed in the population. Okay. But I think what you're kind of getting at is more long-term effects that that we don't know what we're going to be dealing with down the road. And I will tell you that here in the United States, that further safety data on both vaccines will be reported to the Vaccine Adverse Event Reporting System, and that program is an existing national early warning system that was set up to detect possible safety problems in licensed vaccines. But I think but down the road, whether there's going to be any long-term, I think we don't know. We don't know the long-term outcomes of these because they are new. And I think all we can do is utilize the resources that are currently available and reporting mechanisms that if anything does come up, that it is reported, at least in our country, to the CDC and the FDA. But because they are new vaccines, we don't unfortunately know the long-term effects. But I think just one quick, when you talked about the anaphylaxis, like we do know that I think the current estimate is that anaphylaxis will occur at approximately like 1 in 100,000 doses. But I think we also have to remember that this rate of severe allergic reactions, while it might be higher than other vaccines, it's substantially lower than the rate reported for penicillin, right? And penicillin is really popular. So that's actually estimated to be 1 in 5,000. Okay. So already we're seeing that the COVID vaccines, yes, there are going to, we see this with vaccines, that there are these risks of side effects, but we have to look at it from the big picture too, and with what we know about other vaccines, and that it's definitely not within the range of concern when we look at like ones like the penicillin.
Just on this again, I very probably have misunderstood this. I seem to have understood from talking to people that not in the mRNA vaccines, but in the other vaccines, they're using, I can never get the adenovirus. Is it?
Mm-hmm. Adenovirus, adenoviral vector.
Mm-hmm. That these are vaccine. This particular vector has been used for a very long time and very safely. And that because of that we can be reassured of its long-term safety. Is that correct?
Yeah, I'd have to go back and look through some of the. I know we have. I just want to. Because the viral vector vaccines are definitely an existing licensed human vaccine that using the same platform. Right off the top of my head, I'm blanking on which ones, are currently licensed that use this, but it is an established system that has been used before, most definitely.
In my memory, it's from a chimpanzee cold vaccine or something. I remember thinking, oh, that's interesting.
Well, so what it is so that's interesting that you mentioned that. So in this case, like for the so when we start looking at when we start talking the chimpanzee adenovirus, that basically typically causes like harmless colds, but they just use that vector to shuttle the coronavirus gene into human cells to spur an immune response. So like there's the viral vector used to help make the vaccine is from chimpanzee, but chimpanzee cells are not used, and you're not going to get anything chimpanzee in it. So yes, it's going to be safe, but the issue is that like companies like AstraZeneca and Johnson & Johnson that use this adenoviral vector system, a lot of times they do use fetal cell lines you don't have to. I mean, there's definitely examples of adenoviral vector systems that don't need the fetal cell lines, but that's just something you need to kind of look carefully at if you're trying to avoid the use of fetal cell lines.
That's as an ethical concern, certainly.
Right.
Was there, it's gone now. Couple of to that. Some people are concerned that some of these vaccines may contain material other than the vaccines. Can you reassure them that there isn't anything else in these vaccines except what's on the label, as it were?
So you can definitely go.
Shall we say?
Yeah, so there are going to be what's called like excipients, right? So when you start looking at the Moderna and the Pfizer vaccine, I mean, there's just going to be there is going to be the nucleotide. They are encapsulated in what's called like a nanoparticle or a lipid, and that just helps to shuttle the vaccine basically into the cell lines, and it gives it stability. Okay. But I know that there is some concern because there have been reports that it's the fact that these vaccines contain what is called polyethylene glycol. And that's an inactive ingredient. And I know in the Pfizer vaccine, I think it actually might be in the Moderna vaccine as well, but don't quote me on that. I have to double check. So I know that there is concern with this polyethylene glycol being in it and whether people maybe will have an allergic response to that. And that is one of the current leading suspects in causing some of these reactions. But because of these rare events, the administration of the vaccines includes a period of 15 minutes of observation, as you mentioned earlier. But I just want to also emphasize that polyethylene glycol is not anything that we're unused to seeing. So actually several cosmetics and medications contain polyethylene glycol. Some common household over-the-counter pain medications, such as the Tylenol and Advil gel capsules, they actually contain polyethylene glycol. So it's not anything new that we haven't seen before, but it is something to be aware of, that if people have had a bad reaction to that in the past, they need to be but that's why they built in this monitoring time. Yeah.
But shall we say there's nothing more exotic, shall we say? There's nothing in the line of, say, micro silicon in there.
No, there's nothing foreign and no microchips or anything like that. It's all just basic biology tools that are needed to make it stable and that your cells will take it up effectively and that it actually works.
I understand, obviously, you're not an epidemiologist. I don't know if you're a virologist. Are you a virologist?
No, I'm actually a biochemist.
You're a biochemist, okay. But so again, I don't want to put you in a position of having to say, I'm sure you'll say you won't, but in the context of your understanding of the nature of this virus and the nature of the pandemic and its rate, the infection and contagion and its CFR and etc.
Mm-hmm.
Do you think if the vaccine is necessary? I mean, do you think that this is something that people should take seriously about saying, okay, I think I will take this vaccine, whether for their own health or maybe in the consideration of the public good,
Right.
create wider immunity? Is it necessary? Some people have said, you know what, this isn't that important. It's just like a flu. We're all getting too excited. We should just let it go.
Mm-hmm.
Mind people who are, very elderly people should be cocooned, but we should otherwise just, as it were, let it go through the population naturally. Is it necessary, this vaccine?
Yeah, no, I think that's a really valid concern and an excellent question, because I know that some people are wondering, well, what if I just get infected with the natural virus? Isn't that going to be better maybe than getting vaccinated? Or what if I just wait around to see if I haven't gotten it yet? What if I just wait around to see if I do get it and then I won't have to get vaccinated? I just want to maybe back up a little bit and talk scientifically about this. So the goal of vaccination is going to be to teach our immune cells, as we were talking earlier, our T cells and B cells, to teach them what this virus looks like so that they can recognize it quickly and prevent infection and illness in the first place. So we really need this vaccination as a way, or the idea of the vaccination is going to be to train your immune system to respond to the virus without any disease effects. So if you don't get vaccinated, you are putting yourself at risk that you will get the disease. And we know that some of the, or you'll transmit it to somebody and they will get the disease. And we do know that severe disease has resulted in some, hospitalization. It's resulted in death in some cases. And we know from the clinical trials that have been done, especially with Moderna and Pfizer, that they have shown that, and this is the strongest clinical evidence that we have, that the vaccines lowered the chance of developing COVID-19 by around 95% as compared to the placebo injections. Okay, so they prevented not only any disease due to the virus, but severe disease. And so I think that's where every person has to really decide whether you think it's important for you and your family to get vaccinated in order to protect yourself from potentially yourself and your family if you were to transmit it to somebody else, not only from being infected, but to actually acquiring this severe disease, because you might not be protected. And we know that the vaccine does not appear to give permanent immunity. Neither, and it doesn't appear to be the case if you get the disease either. So your antibodies will eventually go down whether you have the disease or whether you got vaccinated. So the whole idea then is, and even people that were previously infected and recovered from COVID-19 are actually advised to receive the vaccine because they know that it will help to keep their immune system bolstered and protect them from being reinfected. So this is a tough decision. People really have to make their own ethical decision and their own choice,
I think a question a lot of people wonder, whatever, okay, it will protect me, maybe it would, strong protection against me developing severe disease. If I'm vaccinated, does that also reduce my capacity to transmit the disease?
I think I think they're still looking carefully at that data. I don't know if it's absolutely clear whether it will actually prevent asymptomatic transmission. I don't think that's clear yet. But I do know that I think it's pretty clear that it will reduce the infection in an individual. And if that occurs, then there is a less likely chance then that you will be contagious, and then you will pass that on to somebody else.
Because it is true, it seems to be the case that with this virus, that if you yourself are infected, but have a very low level of infection and carry a low viral load, you are far less likely to infect. And that there was the number of studies of the most recent is that conservatively they estimated in the United Kingdom that people who are asymptomatic will be at most a third as infectious as people who are symptomatic. So if the vaccine makes you asymptomatic in a sense, then you are at a third less infectious than somebody. So I'm aware of the time, so I want to get to a last issue there. A lot of people, so you've talked about sort of the issues. Assuming that, say, you have access to an ethically produced vaccine, that its production, its trials, etc. I mean, a lot of people are asking, and I have asked me and said, well, would you take it? And my answer is, Frank, if I have access to an ethically produced vaccine, personally, to the extent that anybody cares, I would say, yes, I would take it, and I would recommend members of my family would take it. How, if I can be so presumptuous to ask you yourself personally, so for yourself, for your children, for your family, would you be happy if this vaccine, when it becomes available, to make use of it?
Yes, I mean, if the vaccine becomes available to me and my family, I do plan to get vaccinated. Especially if one becomes available that was not produced with fetal cell lines. That would be my goal, to go out of my way and seek out an alternative. If one was available that didn't have any association with fetal cell lines, especially like testing and development, I mean, I would even go that step further. That would be ideal.
And you could possibly CureVac may be that. Is that?
That looks to be the that looks to be the case, and you can actually find that on our chart. I just want to, because I want to, let's see, give you, yes, because it's a vaccine that comes out of Germany, and so right now I think it's more in the earlier phases, so it's obviously not approved yet. But right now we have not found any confirmation that they have used any fetal cell lines at any of the 3 phases that we have looked at.
And I know that I was looking at the process. They have these progress charts of all the various 97 different vaccines that are in trial at the moment. And CureVac actually has accelerated enormously in the last little while, and they're hoping to go into conditional license to submit their data in February.
That's great.
And so inshallah, as they say, that may be available sooner than we had hoped for. But listen, Dr. Tarasan, thank you again. I suspect this is not the last time we will talk. This is going to be a source of concern and of interest to pro-life people for quite some time. But again, thank you so much for your time. Thank you for the clarity of your answers and your honesty. I'd again refer, anybody should go and have a look at the Charlotte Lozier website. We will be putting a link. It's a great organization and lots of really interesting stuff there. But for tonight, because I know you have a hard closer, I'm going to say thank you very much for joining us, and God bless, and stay safe.
Thank you. You too. Thank you so much. God bless, and look forward to being in touch in the future.
Thank you. Bye-bye.
Bye-bye.
Okay, folks, that was great, I think, from Dr. Sanderly. And again, before we close up, I'd like to thank you for your presence and for your questions. I hope we've helped. And again, I'd say if you hit the subscribe button, please, and if you can help us out on the old donations, anything you give us will be used to the best of our abilities in our projects to defend life and the values of life that we all share. Thank you, and stay safe. Bye-bye, and God bless.